everyone, and welcome to my seminar presentation entitled Magnetic Nanoparticles Stimuli-Responsive Iron Oxide in Battling Resistant Cancers. In this presentation, we will be discussing an overview of cancer resistance, cancer mechanisms of multi-drug resistance, and magnetic nanoparticles with their versatile modes of actions. Then we will take you on to magnetic nanoparticles' common synthetic pathways, in particular the co-precipitation method, magnetic nanoparticles characterization, and finally, applications of magnetic nanoparticles in resistant cancers, where we will be discussing 4 different models. In the end, there will be a graphical abstract and conclusive remarks. As many of us are well aware that cancer resistance continues to be a substantial problem in the clinical setting. As a matter of fact, the multi-drug resistance is responsible for over 90% of deaths in cancer patients receiving traditional chemotherapeutics or novel targeted drugs. Cancer mechanisms of resistance are versatile. A cancer can develop resistance by, for example, an enhanced drug efflux, such as the case with the P-glycoproteins, kicking the chemotherapeutic agents outside of the cancer cells immediately after entry, or by elevated metabolism of the xenobiotics, which is another name for the foreign substances or the chemotherapeutic agent that is introduced into the body. Another mechanism can be through the increase in DNA repair capacity, which indicates an increase in the expression of the DNA repair proteins, making the cancer more resistant to treatment. Next, the growth factors, presenting the associations between the inflammation and cancer development and progression, where acute inflammation promotes the tumor eradication, while chronic immune response leads to tumor growth and invasion due to the increased autocrine production of the growth factors, including different interleukins that were usually observed in the multi-drug resistant cancer cells when you compare them to the drug-sensitive tumor cells. How, you might be wondering? Well, these growth factors can promote the cancer growth and differentiation, but at the same time, some chemotherapeutics, they target the growth factor receptors on the cancer cells. So having an increased secretions of the growth factors lead to competition on these receptors. Finally, genetic factors such as gene mutations, gene amplifications, or gene alteration. Now, in gene mutations, the frequency of lost chromosomes during mitosis are responsible for losing drug-sensitive genes or for changes in biochemical pathways, both of which are crucial in the chemotherapeutic drug resistance. In addition, normal cells, which rarely gain or lose a chromosome, usually stay sensitive to drugs, which makes the treatment even more complex. For gene amplifications, the cancer cells can overcome enzyme inhibition by enhancing transcription of the genes which encode that enzyme. This produces more enzymes because the drug concentration is limited. At some point, it is not able to inhibit the increased amounts of the enzyme. Then, the epigenetic alterations, which has to do with the DNA methylation. Tumor suppressor genes can be silenced by their DNA hypermethylation. On the other hand, the expression of oncogenes can be enhanced by their DNA hypomethylation, which leads to cancer development and resistance. Magnetic nanoparticles are composed of, as the name suggests, a magnetic core, surrounded by a protective coating over which organic linkers can be attached to and carry the active molecules. Several advantages can be attained by utilizing these particles, as the iron oxide in the magnetic core makes them responsive to an external magnetic field. That in addition to their small size privileges and the flexibility in surface modifications to achieve the targeted therapy. But the question here is, how do magnetic nanoparticles overcome resistance when they are loading the same chemotherapeutic agents? The answer lies in their unique synergistic modes of action. Magnetic nanoparticles are stimuli-responsive, and according to their design, we can achieve tumor ablation by magnetic hyperthermia, for instance, where the magnetic particles would heat up in response to a magnetic field causing cellular death. or by photothermal therapy, where the light absorbed by the magnetic nanoparticles is converted to thermal energy causing cellular death, or by photodynamic therapy, where the photosensitizing agents attached to the magnetic nanoparticles are activated by an external light source to create singlet oxygen species that are cytotoxic, or by a mechanical effect where the particles move in response to an external magnetic field, causing cellular rupture. The scheme simplifies the explanation that when we have drug-sensitive cancers, they will be responsive to the therapeutic agent loaded by the magnetic nanoparticles. But when we have a drug-resistant cancer, it is as though they are wearing a mask, and the response to the therapeutic agent is not as efficient. However, the stimuli-induced effect can still lead to their killing. We move on to the synthesis. The most common synthetic pathways are the co-precipitation method, sol-gel method, microemulsion method, hydrothermal method, and thermal decomposition method. The table summarizes the condition being simple or complicated and the corresponding particle size distributions and shape uniformity. For the sake of time, we will only explore the co-precipitation method. Co-precipitation method. A beaker with stirring deionized water is placed on a hot plate. Ferric and ferrous salts are introduced as precursors in fixed ratios, turning the color of the solution into orange. Then, coating agents and ammonium hydroxide base are added. The ammonium hydroxide functions as the precipitating agent and will lead to the formation of iron oxide nanoparticles. The solution then turns black, indicating the end of the reaction. A filtration step is done afterwards and two washing procedures are done. The first wash is done with deionized water to remove chloride ions of the salt, and the second wash is done with ethanol to remove excessive coating. Finally, the solution is dried in a vacuum. Characterization. Magnetic nanoparticles characterization include four main elements. Size and morphology, which can be tested by transition electron microscopy, high-resolution transition electron microscopy, scanning electron microscopy, or dynamic light scattering. Structure and elemental analysis can be tested by energy dispersive X-ray diffraction or X-ray diffraction. Magnetic properties can be tested by superconducting quantum interference device or vibrating sample magnetometry. And finally, surface characterization, which can be tested by X-ray photoelectron spectroscopy, zeta sizer, or Fourier transform infrared spectroscopy. We move on to the applications in resistant cancers. We explained before the additional synergism different stimuli equip our magnetic nanoparticles. Now we will explore their effect more closely in the applications, starting with the mechanical effect. Magnetic nanoparticles' mechanical effect on MCF7 ADR, which is a resistant form of breast cancer cells that usually happens due to the overexpression of P glycoproteins kicking the chemotherapeutic outside of the cells. The figure represented compares the in vitro cell viability of the MCF7, which is the drug-sensitive breast cancer cells, versus the MCF7 ADR, which is the drug-resistant breast cancer cells, when exposed to different treatments that are control, doxorubicin nanoparticles, magnetic nanoparticles alone, and doxorubicin-loaded magnetic nanoparticles, under the influence of no rotating magnetic field, 1 hour rotating magnetic field, and 2 hours rotating magnetic field. So in the first figure, we can see that the control in the MCF7 cells, it had a 100% viability, indicating the absence of a treatment. The pink bars representing magnetic nanoparticles without any drug, we can see that with the absence of a rotating magnetic field, there is no decrease in the cell viability. But with the application of 1 hour rotating magnetic field or 2 hours rotating magnetic field, there is a significant decrease in the cell viability, indicating the mechanical effect even though there is no drug loading. The purple bar representing the doxorubicin magnetic nanoparticles have shown a significant decrease in the cell viability on MCF7. However, this decrease is not selective. It was seen in the absence of a rotating magnetic field and the presence of the rotating magnetic field. This means that the doxorubicin nanoparticles will attack the normal cells as well as the tumor cells in a similar fashion. Finally, the yellow bar that represents the doxorubicin-loaded magnetic nanoparticles, it shows a slight decrease in the cell viability in the absence of a rotating magnetic field, a more significant decrease with the application of a rotating magnetic field for 1 hour, and an even higher decrease with the application of a rotating magnetic field for 2 hours, that is even higher compared to the doxorubicin nanoparticles. The color map translates the same results and emphasizes the selectivity of the magnetic nanoparticles. We can see that in the doxorubicin-loaded magnetic nanoparticles, in the absence of an RMF, the color was green, which indicates a higher cell viability. But as you begin to apply a rotating magnetic field, there is a more significant killing towards the red color, with the highest efficiency being seen with the application of a rotating magnetic field for 2 hours. Similarly enough, in C and D, representing the cell viability for the MCF7-ADR or the resistant cancer cells, we can see that for the magnetic nanoparticles, having no drug has also seen a significant decrease in activity with the application of a rotating magnetic field for 2 hours. The doxorubicin was less efficient in this case because the breast cancer cells are resistant to it. However, when we use doxorubicin-loaded magnetic nanoparticles with a 2 hours rotating magnetic field, it has shown the best decrease in the cell viability. The next synergistic mode of action would be magnetic hyperthermia. Magnetic nanoparticles hyperthermia on lung cancer stem cells, which is a resistant form of lung cancer because it contains stem cells in it that can change and mutate very rapidly. So in figure A, we are looking at the in vitro cell survival rate percentage, comparing different groups of control, heat shock protein, silica nanoparticles, magnetic silica nanoparticles loaded with a heat shock protein, and finally a targeted magnetic nanoparticle that also contains the CD20 antibody. We can clearly see that the best results were obtained with the antibody-loaded magnetic nanoparticles that were subjected to the alternating magnetic field, represented in the white bar. Mechanism-wise, the magnetic nanoparticles not only targeted the tumor site, but were also killing the tumor cells using the hyperthermia that was stimulated by the external magnetic field applied. Figure G displays the iron element biodistribution of the magnetic nanoparticles in mice after 24-hour IV injection. Significant accumulation was found in the tumor site, indicating targeted therapy, and significantly high levels were also found in the liver as the main organ of metabolism and clearance. The third synergistic mode of action would be the photodynamic therapy. Magnetic nanoparticles, photodynamic and pH responsive on MCF7-ADR. This study utilized a dual action of pH sensitivity to target the tumor's acidic microenvironment, and also involved photodynamic therapy induced by laser by including photosensitizing agents within the magnetic nanoparticles to kill the tumor via the production of singlet oxygen species. The first figure shows doxorubicin cumulative release % in vitro over time at different pHs, where the black line at acidic pH displays increased selective release. The second figure shows relative cell viability % of MCF7-ADR resistant breast cancer cells versus the MCF7 sensitive cancer cells treated with different concentrations of free doxorubicin and doxorubicin-loaded magnetic nanoparticles. Significantly higher anti-cancer activity was noted for the magnetic nanoparticles in both the sensitive and the resistant cell lines. The final synergistic mode of action would be the photothermal therapy. Magnetic nanoparticles' photothermal effect on triple-negative breast cancer cells, which are a resistant form of breast cancer that doesn't contain the receptors often targeted by chemotherapies. The first figure shows a bio-inspired magnetotactic bacterium containing naturally present biocompatible magnetic nanoparticle clusters that are genetically produced in specific organelles called the magnetosomes. The researchers extracted these magnetic nanoparticles as photothermal agents for triple-negative breast cancer cells therapy using laser and magnetic field as therapy amplifiers to kill the triple-negative breast cancer cells with just one or a few low-energy laser pulses. This we can see in figure C, which shows the percentage of detected and destroyed breast cancer cells under one laser pulse at different laser energies. And we can see with the increase of the laser energy, there is a more significant killing of the cancer cells. The insert shows optical images of destroyed cancer cells after the laser therapy. This is the graphical abstract summarizing everything that was mentioned in this presentation. We've talked about resistant cancers and their mechanisms of resistance, making them less susceptible to regular chemotherapeutic agents. Then we introduced magnetic nanoparticles as promising agents to battle such resistant cancers. We talked about their synthesis and the co-precipitation method being the simplest and the most common method of synthesis, plus some of the characterization methods. Then we introduced the synergistic modes of action by the application of an external stimuli that makes these magnetic nanoparticles unique, such as the case with hyperthermia, which is the application of an external magnetic field to induce heat, photothermal, which is the application of a light source also to induce heat, photodynamic, which is the application of a light source to induce singlet oxygen species that are cytotoxic, and finally the mechanical effect by applying an external magnetic field to help these particles move and lyse the cancer cell membrane. Thank you for listening.